In the previous video, we have studied about wet sieving method, which is a process of isolation of AM fungi. Now we will see the second method, which is sucrose gradient method. Developed by Daniel and Skipper, 1982, commonly used technique for AM spores extraction, means that this method, sucrose gradient method, is given by Daniel and Skipper in 1982, and this method is commonly used for spores extraction of AM fungi. Requires prior sieving and decanting. Here, before this process, the sieving and decanting method should be done. This gradient centrifugation method is result of many modifications right from Ohm's 1957, Morse and Jones 1968, Mertz et al 1979, etc. This process of centrifugation, gradient centrifugation, is can give many modifications in the process. Spores were purified by resuspending the sieving in the 40% sucrose solution, and centrifugation was carried out. First, the spores are purified, and then the process of centrifugation is led on. Centrifugation was carried out at 1750 rpm for 5 minutes. The supernatant was removed and poured into the sieves. The centrifugation is carried out at 1750 rpm for 5 minutes, and then the supernatant, the liquid in the test tube, is then removed and poured into the sieves. The spores that hold on the sieves are carefully rinsed with tap water. Now the spores which is in the sieve means that it is a net-like structure where the supernatant is poured in it, and this sieve is now which holds the spores of the AM fungi. Now it is rinsed, washed off by tap water. The spores were collected by using dissecting microscope. By the help of dissecting microscope, the spores are collected. Mertz et al. 1979 protocol needs a special mention. They used discontinuous sucrose gradient to recover large number of spores from massive soil samples. The sucrose gradient is recovered in the large number from the soil sample, the spores from the soil sample. Mertz et al. He performed a large number of spore extraction in the sucrose gradient method. They decanted and wet sieved 18 kg. It is a large number, 18 kg of soil with cold water, and found that most spores were present in the 425 to 250 micron meter fraction. Means that he took 18 kg of soil with cold water and performed the method of sucrose gradient, and found that most fraction of the spores are present at 225 to 250 micron meter. Spores were separated from most of the remaining debris using this continuous 30% aqueous sucrose gradient. By the use of 30% aqueous sucrose gradient, he separated the debris, that is waste from the in the test tube. The waste by the process of centrifugation, the spores comes up and the debris are top of that, so that waste material debris is also removed. The sieved material was laid on 600 ml water over 200 ml sucrose in 1 liter beaker. In 1 liter beaker, it was laid at 600 ml and 200 ml of sucrose. After setting, the spores and debris that collected at the interface were removed by vacuum aspiration. By the help of vacuum aspiration, the debris were removed. Rinsed in cold water means washed in cold water and centrifuged for 1 to 5 minutes at 1600 into grams in clinical centrifuge on a second gradient 15 ml of water over 200 ml sucrose in 50 ml tube. The duration of centrifugation being determined by the kind and amount of debris present. The debris were collected and removed by the vacuum aspiration, then rinsed off by cold water and centrifuged at 1 to 5 minutes of centrifugation in the clinical centrifuge, and the second gradient was of 15 ml water over 200 ml sucrose. In 200 ml sucrose there was 15 ml of water in 15 ml of tube. 15 ml of tube was taken and 200 ml of sucrose was put in and 15 ml of water. Now this is the diagram of sucrose gradient centrifugation. Here you can see that this is sucrose 5%, sucrose of 35%, sample in 40% sucrose. It is giving DRM and non-DRM. Now here we can see in the first test tube it is a centrifuge tube filled with density gradient solution. The process is done of centrifugation and now it is filled with density gradient, and here centrifugal force is applied and it is filled of density gradient. Centrifuge tube filled with density gradient solution. The second tube contains sample applied to top of gradient. On the top of gradient, with the sample which is applied, this is the sample zone. And in third test tube we can see that under centrifugal force particles move at different rates depending upon their mass. Mass means weight. Depending upon their weight, we can say that it is separated into three parts. The lightest one on the top, the heaviest in the bottom of the. So now in the next video we will study about mass production of AM inoculants and their field application.